Good morning. Welcome to this bio. Now we're going to discuss about the structure of proteins. Proteins are large biological molecules consisting of one or more long chains of amino acid residues. Once the amino acids are connected in a peptide in a polypeptide with peptide bonds, they are called amino acid residues. Peptide bonds are formed during translation of mRNA in the cell. All the proteins composed of standard amino acids, around 21 amino acids, which are specified by the genetic code. So these standard amino acids are called alpha amino acids, as both the functional groups, amino and carboxyl groups, are attached to the same carbon atom. Only amino acid having secondary amino group is proline. The sequence or the structure of amino acids can be broadly classified into four major categories: primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure, it represents the sequence of amino acids in a peptide. So it is glycine, RNA, serine. It is a part of polypeptide. So these are connected by peptide bonds. Then it is also represented from amino group towards the carboxyl. Here, from amino group towards carboxyl. This is one amino acid. This is another amino acid. This is another amino acid connected by peptide bond. So we represent here from amino group to carboxyl for the sake of convenience. Primary structure stabilized by peptide bonds. Peptide bond is planar. So we can say this is peptide bond. So both the groups of the peptide bond are in the same plane. So it is called planar. And it has partial, this is peptide bond, right? It is represented by partial double bond character. It has partial double bond character. As the result, it has restricted rotation. So unlike the other two bonds, which are shown by pi, psi and pi, these DNs, psi, omega, and pi, these are called torsional angles or dihedral angles. So these two are single bonds. As a result, they are easy to rotate. But here it has partial double bond character. It has restricted rotation, and so as a result, we say it is rigid. And the two, this peptide bond has an angle of 180 degrees. So it forms a straight line. As a result, it is more stable. The two, here, CO and LH groups are in the same plane, and they are trans to each other. As a result, it is more stable. If they are cis in position, it would have more repulsion. As a result, it would be less stable. But here in this case, these two are trans in position. As a result, it is more stable. The other two bonds represented by psi and pi. You can remember the bond in between CR and CY is psi, C, psi. The bond in between C is psi. And the bond in between NH and alpha carbon is represented by pi. These two have a range of values from minus 180 degrees to the plus 180 degrees. So this peptide bond can be shown with another canonical form, showing here the lone pair of electrons on the amino group being transferred to this bond, and the double bond electrons are being transferred here. So as a result, we show it here O minus and N plus. Peptide bond formation is not favorable. That is, it is an endergonic reaction or endothermic reaction that need ATP to form the peptide bond. And peptide bond is also broken by hydrolysis and normal condition in a spontaneously without any requirement of enzyme, but it is a slow process at normal condition. So dihydrolysis are internal degrees of freedom in a polypeptide. They control the protein conformation. Primary structure represented by 3D structure of protein. So post-translation modifications of polypeptide also comes under primary structure. Sequence of amino acids in a polypeptide can be determined by using Edmond degradation and even tandem mass spectrometry. Even a small change in the arrangement of amino acids in a polypeptide can lead to the entire shape change. As a result, its function. See, for example, sickle cell anemia, where beta chain of hemoglobin at sixth position, valine is present instead of glutamate. Glutamate is an acidic amino acid, but valine is a neutral amino acid. As a result, sickle-shaped RBC that clog blood vessels. So that is the whole primary structure. Next, the secondary structure. Secondary structure is stabilized mainly by hydrogen bonds and that are present within the backbone of the polypeptide. And it is formed in a regular local conformation of linear sequence of polypeptides. So hydrogen atom of NH group is hydrogen bonded donor, and the oxygen atom of CO group is hydrogen bond acceptor. So other bonds include peptide bond also stabilize secondary structure. Secondary structure represented in alpha-helix, beta-helical sheet, random file. Alpha-helix, it is characterized by a specific number of amino acid residues per turn. Position of amino acid residues related to its neighbors are also important as the interactions between the side groups of the amino acids may have either repulsion or attraction. That is either stabilize or destabilize of helix formation. So helix may be right-handed, this is clockwise direction, or left-handed, that is anti-clockwise direction. This is right-handed alpha helix, this is left-handed helix. Then, if based on the number of residues in between which hydrogen bond formed, these are classified as gamma helix, beta helix, alpha helix, pi helix. If the hydrogen bond formed in between N and N plus 5 amino acid, then we have pi helix. If the hydrogen bond formed between N and N plus 4 amino acid, that is called alpha helix, which is more stable. And if the hydrogen bond formed in between N and N plus 3 amino acid, then we call it as beta helix. If the hydrogen bond formed in between N and N plus 2 amino acid, then we call it as gamma helix. Then, so we can represent here, that is N amino acid, N plus 1 amino acid, N plus 2 amino acid, N plus 3, N plus 4, N plus 5, N plus 6. So we have represented one this if we have one form in between N amino acid and N plus 3 amino acid, then we call beta helix. If they have one form in between N and N plus 4 amino acid, then we call alpha helix. So this is the helix alpha helix formation. This is alpha helix. So here we have represented one helix. This is also called pitch, has 3.6 residues per turn in alpha helix, specifically alpha helix. And the distance or length of one helix in alpha helix is 0.54 nanometers. Here we have shown that we can say this helix riser, that is the distance between two helices is 0.15 nanometers. Then alpha helix stabilized by some amino acids like malic, M for methionine, A for alanine, L for leucine, E for glutamate, K for lysine. These amino acids stabilize alpha helix formation, and some amino acids like proline and glycine, PC, that is proline and glycine, destabilize the helix formation. Proline destabilize because it forms bend or kink in the polypeptide due to its ring structure. And glycine has more flexibility as it has now any bulky groups at the alpha carbon. So it has more flexibility. As a result, it also destables the helix formation. A long block of glutamate or aspartate. Glutamate and aspartate are negative charge or acidic amino acids, or so these have negative charges. As a result, they have repulsion between them, so they destabilize. Or a long block of lysine and arginine. These two are basic amino acids which have positive charges. Also, these repulse each other. As a result, destabilize the alpha helix formation. Beta plication sheet or beta sheet. It is extremely extended compared to alpha helix. So it can be observed from the diagram. Alpha helix, this is beta sheet. So beta sheet is extremely extended compared to alpha helix. So in beta sheet, polypeptides are arranged side by side, either parallel or anti-parallel. If they are arranged parallel, they form parallel beta plication sheet. If they are arranged anti-parallel, they form anti-parallel beta sheet. So the hydrogen bonds are formed in between two different amino acids of two polypeptides, so which are arranged in the perpendicular to the direction of beta sheet, unlike in the alpha helix. In the case of alpha helix, hydrogen bonds are formed in the direction of polypeptide. But in the case of beta plication sheet, hydrogen bonds are found perpendicular to the direction of the beta bracket sheets. Even in this case also, so this is the direction of polypeptide. This is the direction of hydrogen bonding. So this can be beta bracket sheet can be two types. That is parallel beta bracket sheet and anti-parallel bracket sheet. So in this case, two polypeptides are in the same direction. So it can be represented like this. So here the hydrogen bonds are found in between CO of one polypeptide and the MH of another polypeptide. So these hydrogen bonds are perpendicular to the direction of the polypeptides. Then, so we can say, so the hydrogen bonds here have different angles. They are not straight. They are in different angles. This case, you see, these are straight. These have 180 degrees, but these are not straight. These are in different angles. So these are less stable. As a result, we can say parallel beta bracket sheets are less stable and at the same time less common. So anti-parallel beta sheet. So in anti-parallel beta bracket sheet, two polypeptides arranged in the opposite direction. So see, this is opposite direction. This is N to C. This is N to C. These two are in opposite direction. The hydrogen bonds are found in between NH of one polypeptide and CO of another polypeptide, which are straight. That means these hydrogen bonds are have 180 degrees of angle. As a result, they are more 180 degrees. That is straight line. They form very straight hydrogen bonding. As a result, it is more stable. So we can say for anti-parallel beta bracket sheet is more stable at the same time more common. Why? It has no regular secondary structure. It is formed by polypeptides that are formed by less number of amino acids, around 4 to 20 amino acids. It has not any regular hydrogen bonding pattern. It is generally formed at the terminal ends of the polypeptide, and it contains large amino acids like serine and even the charged amino acids like glutamate and aspartate. This ranophyte is flexibility and used in intermediate shell formation. So ranophyte has two structures, turn and root. Turn is formed by polypeptide having 4 to 5 amino acid residue. So it connects the polypeptides where direction change of the polypeptide needed. So this is one alpha ion, this is another alpha ion. These two are regular secondary structures which are connected by turn, which is not that much regular. Then that is a ranophyte can be of different types based on the number of amino acids that form the turn. So if the turn formed by N between N and N plus 1 amino acid, then the turn formed is delta. If it is formed between N and N plus 2 amino acids, then it's called the delta gamma turn. If it is formed between N and N plus 2 amino acids, then it forms is beta turn, which is more stable. If it is formed between N and N plus 4 amino acids, then it's called alpha turn. If it is formed in between N and N plus 2 amino acids, it is called pi turn. So beta turn is more stable. If you even see the structure of beta turn, that is N plus 1 N plus 10. The structure formed between N and N plus 3, which is straight line, that is one, that is more stable. So the other two are having some different angles. So other type of ranophyte is blue. That is, it is formed by 7 to contributed with 7 to 10 amino acid. So it forms irregular structure that connect the regular structure like this. These two are different beta sheets which are connected by test structure representing completely for the protein. It is stabilized mainly by hydrophobic interaction of the side chain for amino acids within a polypeptide. Other bonds that stabilize tissue structure include hydrogen bonding or peptide bonds, van der Waals interactions, ionic bonds, covalent bonds. And it is the restriction structure is found in the single polypeptide where polar amino acids reside on the surface and non-polar hydrophobic amino acids reside within the folded protein, that is inside of the folded protein. So we have represented here primary structure, secondary structure, and tertiary structure. Primary structure is shown with the number of amino acids, where M stands for methionine, A stands for alanine, L stands for leucine, E stands for glutamate, K stands for lysine, W stands for tryptophan, Y stands for tyrosine, F for phenylalanine, P for bromine, G for glycine. So these amino acids stabilize the formation of helix, and these amino acids stabilize the formation of beta sheet, and the proline glycine stabilize the formation of turn. So this is secondary structure. For proper fold, use the texture structure. That is example. That is myoglobin is an example for texture structure. So hydrogen bonds are found in between in a polypeptide of texture structure. That is here it is we have shown over the side on example for amino acids. So if the side groups are alcoholic groups, so the amino acids may be serine, tyrosine, and threonine. Alcohol and acid, the hydrogen formed between alcohol and acid. So alcohol, the examples may be tyrosine and aspartate. So acid contains is aspartate, is connected to it. So alcohol and amine, that is alcohol, serine containing alcohol, and the lysine has amine. Alcohol and amine, that is alcohol serine group. Serine has alcohol group, sodium, and amine is present in aspartate. Antibond formation, antibonds are also called alpha-bridges. It is the result of the neutralization of acid and amine groups of the amino acids in a polypeptide. Hydrophobic interactions, these are the main cause of stabilized tertiary structure. It is formed between the side chains of the amino acids called polypeptide, and it is based on the solubility rule, the light and dissolves light. And hydrophobic or non-polar groups generally present inside of the folded protein. And covalent bonds, these are formed between thione groups of cysteine, that is cysteine having SH group is called thione group. So it is formed in between the two cysteine groups. So these bonds are very strong. And the cysteine having SH group is called thione, the other cysteine having another thione. So upon oxidation use disulfide bridge. So it can be present in a polypeptide. This is one disulfide bridge, this is another disulfide bridge. The van der Waals forces, these exist between any groups in a polypeptide and determination of texture structure, X-ray crystallography, NMR, and dual fluorescence interferometer. These three techniques generally used to determine the structure of protein. So X-ray crystallography, it is the most common tool used in determination of the polypeptide structure of proteins, and it provides high resolution about the structure, but it doesn't give you much information about the flexibility, that is conformational flexibility that takes place in a polypeptide due to the environmental changes. And NMR, nucleocalcium resonance, comparatively gives less information about the resolution of the structure, and gives much information about the conformational flexibility that takes place due to the environmental changes. And it is limited to small proteins having around 100 amino acids. And dual interferometry, it provides complementary information about the surface captured proteins, and it determines the structure and conformational flexibility within the polypeptide. Quantum structure proteins, multiple polymers come together and form quantum structure. The main force that stabilizes the quantum structure is the ionic interactions in between the polypeptides, and other uses that include peptide bonds, hydrogen bonds, van der Waals interactions, covalent bonds also stabilize quantum structure. So quantum structure then we contain many polypeptides where different polypeptides have different functions. So we have taken hemoglobin as an example for quantum structure. Here, the four polypeptides of tissue structure form one molecule of hemoglobin, that is quantum structure. Hemoglobin has four polypeptides: alpha 1, alpha 2, beta 1, beta 2. Here we have shown dot as the P plus 2, which is present in the, and this round is shown as C. So this totally this total is called as hemoglobin. That is quantum structure. So in hemoglobin, there are four polypeptides. As a result, they can solve different functions like carrying oxygen and CO2 by different polypeptides, and the holoenzyme contain many polypeptides. So different polypeptides solve different functions in a holoenzyme, like one polypeptide act as regulatory subunit while other act as catalytic subunit. Symmetry in quantum structure. So simplest symmetry that is present in quantum structure is rotational axis of symmetry, and individual subunits can be superimposed, that is brought into coincidence on each other identical subunits by rotation about one or more rotational axis. So if the required rotation is 180 degrees, we can say that is 360 by 2. We can say that the protein has two quantities of symmetry. If the required rotation is 100 degrees, that is 360 by 3, that is we can say that protein has three quantities of symmetry. So we can take a screenshot of it. Thank you.